Hello to our friends joining us via recording. Today we are continuing our work on lesson number 10 about the way that neurons work. To help us get ready for continuing this discussion, we did a little bit of lab review. So over here on the right side, we have a list of several of the important structures of a neuron that we talked about in lab and that are going to be important again here in lecture. We are trying to match up our functions with the names of these structures. So for my friends who are here in class, let's go ahead and grab that pencil up above here. We're going to make our slide super messy, but do me a favor and let's do some connecting here between my descriptions and my definitions. My names and my definitions, excuse me. Let's match up our words and their descriptions. By the way, for those of you who were here that day that we cracked the Star Wars joke, right, about Palpatine, I told my husband that, and he did get a kick out of that we cracked that joke in our class together. So if you were there that day, my husband appreciated that joke. Okay, so looks like we're all matched up. Let's go through and chat about these things really fast. So the first part of a neuron, when we talk about neuron signaling, that's going to be most important are these parts called the dendrites. So think about dendrites like receiver dishes. They are listening for messages. Those messages come from another neuron. To use a word from last class, when we're talking about the dendrites, we are talking about a structure that we find on the postsynaptic neuron, a postsynaptic neuron, which is the one that receives a message, neuron number 2, if you will. So the part of neuron number 2, my postsynaptic neuron that receives a message, is the part called the dendrites. That message is coming from the thing right above it on the list called the axon terminals. So the axon terminals are found on the presynaptic neuron. This is the end of the road for a presynaptic neuron. When it's ready to talk to its neighbor, this is where it's going to spit out those neurotransmitters. And the neurotransmitters are what's actually going to send a message to the neighboring cell. Before a message can get to the axon terminal, it has to travel down the axon. So the axon, if you remember from a neuron, is the really long part. That's what a neuron uses to send a message really quickly. Fun fact about axons, the cells in your body, the neurons in your body that have the longest axons are the ones that go from your spinal cord all the way down to your foot. There's actually single neurons. So there's one neuron, well, multiple neurons, but consider a line of really, really long neurons that go all the way from the spinal cord in your lower back all the way down to the bottom of your feet. Neurons are can be some of the biggest or longest cells in the body. So they've got these really long axons that move the messages away. Those axons, if they're sending a message, especially that far, right? If they're going all the way down the back of your leg, we're going to need to help that message to go quickly and help that message to stay strong. That's the job of the myelin sheath that wraps around an axon to help that message stay strong. For the most part, during our class today, when we're talking about the myelin sheath, we're going to be talking about Schwann cells. So Schwann cells are one type of cell that make myelin sheaths around neurons. Schwann cells are found in a place called the peripheral nervous system. Probably the best way to describe the peripheral nervous system is that it's everything that's not the central nervous system. I know we talked about this when we did this stuff in lab. Can anyone remind me what the structures are that are in the central nervous system? What are the structures in the central nervous system? There's just 2 of them. Yeah, Jesse's totally right there. So the 2 structures in the central nervous system are the brain and the spinal cord, the brain and the spinal cord. Those are the only things that are found in the central nervous system. So most of the nervous system in your body, which is all of those little nerves that go out to your fingers, the stuff that goes from your spinal cord down to your foot, all of that's considered the peripheral nervous system. So the peripheral nervous system, we use a type of cell called Schwann cells to make the myelin sheath. Remember from lab that when I'm in the central nervous system, I won't make you spell these because these are hard. When I'm in the central nervous system, I use a type of cell called oligodendrocytes to do the same thing. So oligodendrocytes make myelin sheaths, so do Schwann cells. Schwann cells found wrapping around the nerves in your body that go all kinds of places. Oligodendrocytes found in your brain and your spinal cord. But the goal is still the same, making those myelin sheaths. And remember that there are little small spaces called the nodes of Ranvier, where we are in between the parts of the myelin sheath. Later today, most likely, we're going to talk about the way that neurons send messages, and they just have to exchange ions with their environment only at the nodes of Ranvier, not everywhere. So this is what we're going to use in the process of neuron signaling. We're just going to jump between these little nodes. So we talked about their functions. Now it's time for us to label their locations on a neuron. So let me start by highlighting this area right here. This long area right here on my neuron, what's that part called? What did we just label there? Yeah, that long part right there is the axon. So that's the part when we were talking about the huge neurons in your body. This is the part that's super, super long. So on my axon here then, I'm seeing a myelin sheath. What did we say since we're in the peripheral nervous system? What's the name of the cells that make these myelin sheaths? Let's draw little lines for them. Yeah, these are all of my Schwann cells. Schwann cells. So these are individual cells. Their one goal in life is to wrap around this little tiny area on the axon, just take care of this small space. You can see in between them, and that's why I pulled this particular cartoon, you can see those nodes of Ranvier really well. So see all those little areas I circled? We'll draw lines up here for us. All of those little spaces in between them are the nodes of Ranvier. And those nodes of Ranvier are going to help us to send our messages. My messages have to go all the way down here. What are these little things down here called? Yeah, absolutely. We're at the end of the axon. So these are the axon terminals. So the end of the axon, or where the axon terminates, those are all my axon terminals. And if we were looking at this neuron in the context of another neuron, if it was talking to another one, each of these little axon terminals that I see right here would be touching the dendrites, which are all of these up here, the dendrites of another neuron. So the last structure for us to label all of these branches that we see here coming off of the cell body of this neuron, all of these are the dendrites. Well, so to be fair, yeah, Nicole asked, do they touch? They don't technically touch. You are correct. There's that little space in between them. The space that would be between the axon terminal, the dendrite, it's called the synaptic cleft. Synaptic cleft. So there's a small space, and we'll see it better on our pictures that you have in your notes. So here, let me try to freehand it here. There's an axon terminal. Here is part of a dendrite, the membrane of a dendrite right here. So they don't technically touch. They're really close to each other. There's just a small synaptic cleft. So what I would see here, let's add it to the picture here. When I'm looking at it from a distance, it kind of looks like they're touching, but they aren't actually physically touching each other. There is a space in between them. So we would see axon terminals from another neuron that lived up here. They're really close to these dendrites here. They don't actually touch. There is a space in between them, and that space is called the synaptic cleft. Yeah, so good clarifying question. Jacqueline asked a question that we'll probably talk a little bit more about today, but I want to put it to the class. Jacqueline asked if neurotransmitters actually go into the postsynaptic cell. Does anyone happen to know? So this cell releases neurotransmitters to talk to this one here. Yeah, when neurons talk to each other, the neurotransmitters that they send to each other, just like we saw when a neuron talked to a muscle cell, my neurotransmitters never actually go into the cell I'm talking to. They stay on the outside and help to open up some of my ion channels. So neurotransmitters always stay in this place called the synaptic cleft, in the space in between them. We do have some little receptors that would be on the membrane of the postsynaptic cell. That's where the neurotransmitters attach, but they're always going to stay outside the cells. They're never actually going to go in these two cells. So the question is, would there be a presynaptic cleft and a postsynaptic cleft? No, there's not two different clefts. There's just one space. The other way we could call the synaptic cleft, the way we were referring to it last time, sometimes it's just called the synapse. So there's just one synapse, or in other words, there's just one synaptic cleft, one space in between them. If you're in front of the synapse or in front of the synaptic cleft, you're the presynaptic neuron. If you are after the synapse, you'd be the postsynaptic neuron. So just one cleft, one space in between them. Yeah, good clarifying question. All right, one last thing that I had us do was work on labeling our graph here. I know I heard at least one of my groups mention this, that at least this graph, it's not the first time you've seen it. We saw it way back in Unit 1, so hopefully we learned a little bit about it in Unit 1, and we'll just keep reviewing it here in Unit 3. We are looking at a graph of the membrane potential of a neuron. Remind me in the chat, this word potential, what was the easy word I told you to replace it with? Yeah, exactly. So this is a graph that technically says it's membrane potential. Membrane potential is, the easy word for it is membrane charge. Membrane charge. So we are looking at all the ways that a neuron changes its membrane charge from when it's not hearing any messages, to when it's hearing a message or talking to its neighbor, to when it's trying to get back to normal. When I talk about the normal charge on a neuron, what is the word that I use to describe what the normal charge of a neuron's membrane is? What's that word for a normal charge? Yeah, it's really long, right? It's the resting membrane potential. I'm going to abbreviate it because I'm feeling a little lazy this morning. I'm going to be real. Resting membrane potential. That's the normal charge on the membrane of a cell. So in the case of a neuron, remember that this was one of the numbers we had to know. We had to know that a neuron normally chills out at -70 millivolts. That's its resting membrane potential. Before it hears a message, and I'll point out after it's done hearing a message, so we need to add a little tag over here too. Whenever a neuron is at -70 millivolts, we would say it's at its resting membrane potential. This is when it's listening to hear a message. When it hears a message, if it decides that that message means it should talk to its neighbor, what's the charge on its membrane when it decides to talk to its neighbor? Where on this graph does a neuron talk to its neighbor? Okay, so I asked my question poorly. I can see where we got there though. A bunch of us are honing here on -55. What is the name of -55? What's this called right here? -55. Yeah, -55 is called threshold. Threshold. Okay. So threshold is the value, and again, based on the way I phrased my question, you're totally right. Threshold is the value when a neuron's membrane hits threshold. When we get to -55, this is where my neuron realizes that it's serious enough for us to get ready to talk to a neighbor. When we hit threshold, we're far enough from resting membrane potential that I'm going to need to talk to my neighbor. So I hit threshold, that shoots me all the way up here to +30. +30 is the voltage or the charge on my membrane where I actually spit out neurotransmitters. So up here at +30, this is when one neuron is going to talk to its neighboring neuron. So I'll put a little note up here, neuron talks to neighbor. That happens here at the top when I get to a charge of +30. The only way I can get to a charge of +30 is first, I've got to get to this threshold value. Once I get to this threshold value, I go through this process I see right here. What's this process right here called? Did we label this part of the graph over here? Yeah, this part of the graph where my membrane charge is going up, or it's getting higher, that's called depolarization. Depolarization, going away from my normal polarity, which is -70. So I depolarize my membrane all the way up to +30, which is the charge that I can talk to my neighbor at. Once I've talked to my neighbor, though, I want to get negative again. What do I call it when I'm working on getting negative again? What's this part of my graph called? I'm working on getting negative again. Yeah, absolutely right. When I'm trying to go back to normal, that's called repolarization. So repolarization is when I'm trying to get back to my normal polarity. I'm going more negative. But remember, we said that with neurons, they're so excited to repolarize their membrane that they actually get too polarized. They go extra negative. What was the name of the extra negative down here? I got a little too excited about my polarization. Yeah, exactly. Hyperpolarization. Hyperpolarization, when I am more negative than I was supposed to be. Sure, I was trying to get negative again. I wasn't trying to get that negative. I am too much polarized. Remember, hyper was too much. So hyperpolarization, I'm too polarized. Over time, I will get back to my resting membrane potential. And I actually heard some of my groups talking about this. When we're talking about going from hyperpolarization, from too negative, to get back to resting membrane potential, we mentioned this briefly last time. Which kind of channels help me to slowly get back to resting membrane potential? Does anyone remember? Yeah, so the type of channels that help me to slowly get back to resting membrane potential, when I go too negative, I'm going to use those leakage channels to kind of leak back to normal. So in the process of repolarization, what happened is I spit out way too many potassiums. Here in depolarization, I brought in way too many sodiums. Slowly, my leakage channels are going to get us back to our normal levels. The other kind of channel that we mentioned that's going to help us get back to normal, I guess it's not technically a channel, to be fair, it's a carrier protein. The sodium-potassium pump. These two things work together to get my membrane charge back to normal. So we leak positives and negatives, and then we get those sodiums and potassiums where they're supposed to be. That's what's going on right here in my shoot back up from being hyperpolarized. Now, several of my groups or both of my groups were working on talking about the types of channels that are are responsible for each of these parts of my graphs here. Both of my groups correctly identified which kind of channel does the process of depolarization. Who does depolarization? Whose job is this? Depolarization. Yeah, so depolarization is caused by voltage-gated sodium channels. Voltage-gated sodium channels. Here, here before I move it all over the place. When we talk about voltage-gated sodium channels, not a trick question, how do voltage-gated channels open? What opens a voltage-gated channel? Yeah, it's all about charge, right? So a charge is what opens a voltage-gated sodium channel. How do I get these voltage-gated sodium channels open? What charge do they open at? Yeah, so they open at -55, and Jacqueline's totally right. What's going to open them, we'll do this in two pieces here. Voltage-gated sodium channels open at threshold. So when I hit -55, that opens up a voltage-gated sodium channel. To get to -55, to get to threshold, I have to use a type of channel that's called chemically gated sodium channels. Chemically gated sodium channels. Chemically gated sodium channels, what causes this kind of channel to open? What opens a chemically gated sodium channel? Exactly. Yep. So neurotransmitters. 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 Oh, that arrow's bad. Let me move it. Neurotransmitters attach to chemically gated sodium channels. This process of a neurotransmitter attaching to a chemically gated sodium channel, that's what starts right here. It's kind of continuing in this area right here. As a teaser for today, we're going to talk about how not all of my neurotransmitters open up sodium channels, but everything that's happening inside here, this is all chemically gated. This is all about the neurotransmitters. So from this point here, on one version of your graph, it says stimulus received. From this point where a neuron first gets neurotransmitters to the point where it gets to threshold, all of these changes in membrane charge, the first 15 millivolts, is all about the neurotransmitters. It's all about the chemically gated channels. If I open up enough channels and I bring in enough sodium to make my membrane charge -55, if I get there, my voltage-gated sodium channels take over. And they're going to completely depolarize my membrane, and it all the way up to +30. Now at +30, I realize that I am the wrong charge. To get me back to the right charge, which kind of channels do I use over here? Who gets me back to the right charge? Yeah, exactly. Who's going to get me back to the right charge is the voltage-gated potassium channels. Voltage-gated potassium channels. Again, same question, second time. What opens a voltage-gated channel? Yep, same answer, right? Voltage-gated channels open at a charge, a certain charge. Voltage-gated potassium channels open because I hit +30. When I hit +30 up here, suddenly I'm way too positive, and my cell knows it can use potassium channels, voltage-gated potassium channels, to get us back to normal. So this process here, the process of repolarization, that's thanks to voltage-gated potassium channels. This process of rapid depolarization, that's voltage-gated sodium channels. To get to threshold, I've got to use chemically gated sodium channels because all of those neurotransmitters help me to figure out whether or not I'm supposed to talk to my neighbor. So again, you all took it a step further than I needed you to. But this is perfect because you actually have this graph where I ask you to label those channels in your packet. What page is that? Is it like page 12 or something? Can someone confirm for me? I really should pull that packet back up. Let's see. Okay, Tierney's saying she thinks it's on page 12. Yeah, so page 12, we just worked ahead. I only told you today to work up through further like page 10, right? So we just did the activity on page 12. Look at you, you're ahead of the game. So that's our big picture membrane potential graph. We've now talked about it twice, so hopefully that'll help us out as we move forward. Before I move into EPSPs and IPSPs, so postsynaptic potentials, any last minute questions about this graph right now? Or give me an emoji. We didn't do emojis today. You know what we didn't do yesterday? We didn't do or last time we didn't do a penguin. Yeah, Hammond's got a question. Go for it.
Sorry if you already went over this, but did you talk about where exactly the chloride channels would go?
So we haven't talked about those yet. We're actually getting ready to though. So we'll label this graph again together. The chloride channels are chemically gated. So they're actually going to be found in the same place that the chemically gated sodium channels are. So they would also contribute to here. But yeah, we'll definitely be talking about them more moving forward. Oh, Nicole's got a cold, it looks like. Yeah, so Christina asked about the other kind of channel that would also be on this graph. The other kind of channel, and again, we'll talk about this some more soon, is called voltage-gated calcium channels. The goal of a voltage-gated calcium channel is to help a neuron to spit out neurotransmitters. So based on the fact that a voltage-gated calcium channel helps a neuron to spit out neurotransmitters, do we have any guesses where on this graph I might see voltage-gated calcium channels activated? Where might this neuron be spitting out neurotransmitters? Well, resting, it's listening. So it's not going to send any messages if it's listening. I promise it's not a trick question. Where did we say that a neuron was talking to its neighbor? Where on this graph does it talk to its neighbor? Yeah, all the way up here at positive 30. So positive 30 is also, in addition to helping me open voltage-gated potassium channels to get my charge back normal, the other thing I'm going to open up here at positive 30 is voltage-gated calcium channels. The voltage-gated calcium channels do nothing for my membrane charge. They are all about getting me to spit out neurotransmitters. So again, we'll talk more about that with some pictures coming soon, but the last kind of channel that we hadn't labeled on our graph was those voltage-gated calcium channels. They're up here at the top as well. So Jacqueline, it's not that it doesn't make a difference. It doesn't make a difference for the membrane charge, but it absolutely makes a difference when we're thinking about the way that neurons talk to each other. So neurons spit out neurotransmitters to talk to each other. If a neuron can't spit out a neurotransmitter, then it can't talk to each other. So I'll show you a picture. Calcium rushes into an axon terminal. When calcium comes in, neurotransmitters spit out. Neurotransmitters and calcium, they can't mix. It's like when someone you really don't like shows up at a party and you're like, okay, I'm done. I'm leaving. That's what happens in the axon terminal. Calcium rushes in, neurotransmitters are like, peace out, we're gone. So they don't have to do with membrane charge, but without calcium, I wouldn't be able to spit out a neurotransmitter. I've got to have calcium to be able to spit out my neurotransmitters. Yeah, so here, let's do a picture to address Christina's question here. Let's look at a picture. I am going to pull up one of the pictures that we left off with toward the end of class last time. What page in your notes is this picture on? Can anyone help me out? Okay, this is on page 5. Perfect. We're looking at a picture of a chemically gated channel. A chemically gated channel. Chemically gated channels, remember, use a chemical key to open up, to get things in. When we're talking about that first part of the graph, here, I'm going to freehand my graph over here. There's that graph. Okay. When we're talking about the first part of the graph right here, the part we talked about relying on chemically gated ion channels, when we talk about chemically gated ion channels changing the membrane charge, what's happening is one neuron that lives up here is spitting out these chemical messages to talk to the second neuron that lives right here. When those messages attach to the ion channel, ions can rush in or ions can rush out, depending on what kind of channel it is. On neuron membranes, we talked about this last time, we've got 3 kinds of chemically gated channels. The first kind of chemically gated channels is the one that we labeled on our graph before, chemically gated sodium channels. Chemically gated sodium channels. We also, though, on a neuron's membrane, have chemically gated potassium channels, and we have chemically gated chloride channels. All 3 of these kinds of chemically gated channels look just like this. They have a place for chemicals to attach to them so that we can open up a space for ions to rush in or out. But here's where that salty banana is going to be important for me. When we talk about sodium, and I open up a channel for sodium, is that going to come in or go out? What way does sodium move? Yeah, sodium's going to go in, so it enters the cell. Which other thing enters the cell? Potassium or chloride? Who else comes into the cell? Yeah, chloride also enters the cell. Okay, 2 things that enter the cell. So potassium, there's a ton of that on the inside already. So potassium leaves the cell. Okay, let's pause for a moment, make sure we're tracking. Chemically gated channels always transport stuff from where there's a lot of it to a little of it. So some places on my dendrites membrane, my receiving part of the neuron's membrane, some places have chemically gated sodium channels. When the right neurotransmitter attaches to them, sodium rushes in. Some parts of my membrane have chemically gated potassium channels. When the right neurotransmitter attaches to them, potassium is going to leave the cell. And some parts of my membrane have chemically gated chloride channels. When neurotransmitters attach to these, chloride's going to rush into the cell. The effects of the movements of these different ions here is going to be different because they each have different charges. When sodium rushes into the cell, is it going to depolarize or hyperpolarize my membrane? What's going to happen to my charge when sodium goes in? Yeah, sodium is going to depolarize membrane, which means it's going to become more positive. The word depolarize means we're becoming more positive. When chloride goes into the cell, chloride has a negative charge. So is chloride going to depolarize or hyperpolarize the membrane? Chloride going to depolarize or hyperpolarize the membrane? Yeah, exactly. Chloride's going to hyperpolarize the membrane because when chloride comes in with its negative charge, inside the membrane becomes more negative. When I attach a chemical to a channel that its job is to let chloride inside, in comes chloride with its negative charges, makes the inside more negative. Hey, we also have chemically gated potassium channels, and their job is to let potassium out of the cell. When potassium leaves, does that depolarize or hyperpolarize the membrane? When potassium leaves. Any ideas? Yeah, totally right. When potassium leaves, we're hyperpolarizing the membrane. We're making the membrane more negative. Okay. This right here, I apologize, green was a bad color to pick, but this right here, we need to underline, highlight, star. Yes, we need to snap a picture of this. We need to know not only the direction that ions are moving, either into the cell or out of the cell, we also need to know what effect that has on the membrane charge. Does it make it more positive or does it make it more negative? These are things we must know because when we're looking at what happens to a neuron's charge, there are two things that can happen when a chemically gated ion channel is opened. Either a chemically gated ion channel is going to make my membrane charge go up, or it's going to make my membrane charge go down. To address Nicole's question, let's use these pictures to get to it. My first picture over here, I'm looking at a change in my membrane charge, made the membrane more positive. What did we say was the word for making the charge more positive? What was that word? Yeah, there we go. What I'm seeing on the left is the process of depolarization. Very small little bit of depolarization, right? We didn't go up very much, but we went up a little bit. My membrane charge goes up at all. I call that depolarization. Notice that my membrane charge over here has gone down, and I've actually gone below threshold in this case. When I get more negative than usual, the name of that process is hyperpolarization. Hyperpolarization. When we use the word, let's add this word to the top just for a reference, because Nicole asked about repolarization. Repolarization is what a neuron does when its charge got way more positive than it was supposed to be. So the process of repolarization is what I use to go from positive 30 back down to negative 70. Now, after negative 70, I keep going more negative. I hyperpolarize things. But the word repolarization is specifically what we're talking about when I'm going back to normal. We don't use the word repolarization when we're talking about what neurotransmitters do, because neurotransmitters, when they open those chemically gated channels, it's not about getting us back to normal. It's all about whether or not we're going to hit threshold. Now, repolarization and hyperpolarization, do both mean that the membrane charge gets more negative. So if it helps you to understand that repolarization and hyperpolarization are both making the membrane more negative, consider them as pretty similar to each other. But instead of using the word repolarization when I'm talking about what happens with a chemically gated channel, this is the only word that I'd use to describe what happens with a chemically gated channel. Either we're doing depolarization, where we get more positive, or we're doing hyperpolarization, where we get more negative. How do we feel about that question? Or what new questions do we have? So I'll add a quick note. Hyperpolarization, make more negative. Depolarization, make more positive. Yeah, so Nicole's bringing us into the discussion that I wanted to have next with these ion channels here. When we were talking about the effects that ions can have on membrane charge, we did say that chemically gated ion channels for potassium, chemically gated potassium channels, made the membrane charge more negative. So when we're thinking about who can do hyperpolarization, chemically gated potassium channels would be something that could be used to hyperpolarize the membrane. There was one other kind of ion channel that we also said, another chemically gated ion channel that we also said made things more negative. Who else can make things more negative? What other kind of chemically gated channel makes things more negative? Yeah, so a couple of votes coming in there. Chemically gated chloride channels, that's the other kind of channel that can lead us to make the membrane charge more negative. Both of these types of ion channels, if I spit out a neurotransmitter, if I release a chemical that opens a potassium channel, that's going to make the membrane charge go down. If I spit out a neurotransmitter that attaches to a chloride channel, that's also going to make the membrane charge go down. Both of those types of neurotransmitters that open up these kinds of channels, they're causing hyperpolarization. They're making the membrane charge get more negative, hyperpolarization. I actually only have one kind of channel that does depolarization. What's the kind of channel that we talked about, the chemically gated channel that does depolarization? Yeah, chemically gated sodium channels. The only kind of channel I can open if I want to depolarize the membrane is a chemically gated sodium channel. The only way I can make my membrane charge go up is by opening up a channel for sodium. And then sodium can rush inside, and if we get high enough, voltage-gated sodium channels will open too. All right, let's pause and think big picture here for a moment. We've looked at the two effects on membrane charge that I can have when a neurotransmitter from one neuron talks to another neuron. Here, let's do my ugly neuron drawings for us, because we need those today. There's neuron number 1. There's neuron number 2. We are looking at the charge on the membrane of neuron number 2 right here. So let's say that neuron number 1 spits out a neurotransmitter to neuron number 2 that opens up a chemically gated sodium channel. Let's say that we spit out a neurotransmitter that comes down here on a dendrite and it attaches to a chemically gated sodium channel. If that happens, the membrane charge on this neuron is going to go up a little bit. And it going up a little bit is going to get me closer to threshold. Maybe it'll be enough to get me to talk to my neighbor. Maybe it won't. We don't know. So my neuron over here spits out a neurotransmitter that attaches to a chemically gated sodium channel. That makes the membrane charge on this neuron get more positive. Maybe instead of spitting out a chemically gated or a chemical that attaches to a chemically gated sodium channel, maybe instead that neuron spits out a neurotransmitter that opens a chemically gated potassium channel. So I spit out a neurotransmitter, it comes over here and attaches to a chemically gated potassium channel. That neurotransmitter is going to make my membrane charge get more negative. Because remember, potassium leaves a cell and makes the cell more negative. So if I spit out a chemical key that opens up a potassium channel, my membrane charge goes down. So membrane charge down because we opened up a chemically gated potassium channel. Same deal happens if this neuron spits out a neurotransmitter that attaches to a chemically gated chloride channel. That's also going to make my membrane charge get more negative. So when we think about these chemicals, these neurotransmitters, either they're going to lead to the process of depolarization if they attach to a chemically gated sodium channel, or that neurotransmitter is going to lead to hyperpolarization if it attaches to a chemically gated potassium or a chemically gated chloride channel. So those chemical keys, really important. Not only do they only open a particular type of ion channel, they also have a particular kind of effect on the charge on the membrane. I have just talked a ton, so I need to pause and I need to give us a chance to process and to think about what questions we have. So let me pause for a moment and let some questions come into my chat. So Nicole asked a question about voltage-gated channels when my charge gets low. We don't have any types of voltage-gated channels that we're studying this semester that are activated at a lower voltage than normal. And so that's actually kind of the challenge of these kinds of chemically gated channels opening, is when they make my membrane charge more negative, they're making it harder and harder for those voltage-gated sodium channels to open. Hyperpolarization, we could kind of think about this as inhibition, or we're preventing a neuron from talking to its neighbors. When I open up these kinds of channels, a membrane charge gets too negative, the voltage-gated sodium channels have a much harder time getting open. Any other questions we've thought of? Which page do we have this on? Can you guys tell me? Okay, this is page 6. Okay. These are two really helpful pictures to summarize what we just talked about. We just talked about how a chemically gated sodium channel allows sodium to rush inside the cell. A chemically gated sodium channel makes my membrane charge go up because I'm bringing in sodium. It gets more positive. We just talked about how chemically gated potassium channels and chemically gated chloride channels make my membrane charge go down. That makes it harder for me to get to threshold. Now, I have some words, or more accurately, we're gonna go by the abbreviations for these words to describe what happens to the membrane charge when I open these different kinds of channels. First long set of words: excitatory postsynaptic potential. Excitatory postsynaptic potential. That's crazy and long, but we're going to break it into pieces here. You've already told me today what the easy word for potential is. What's the easy word for potential? What could I say instead of potential? Yeah, the easy word for potential is charge. Okay? When I use this word postsynaptic, does postsynaptic mean I'm before or after the synapse? Postsynaptic, before or after? Yeah, post means after, right? So we are after the synapse, or in other words, what we might call this when we're looking at two neurons talking to each other, we might call this neuron number 2. Neuron number 2 is the postsynaptic neuron. This word excitatory right here, excitatory means we're making our membrane charge more positive. We are getting excited to talk to our neighbor. So membrane charge plus, membrane charge more positive. Excitatory postsynaptic potential. Excitatory, we're getting more positive. Postsynaptic, in neuron number 2, my receiving neuron, and we're talking about its charge. An EPSP, this is what I think you should study it as, because I'm never going to ask you to write for me what that stands for. An EPSP is a change in membrane charge that's positive. So I got a neurotransmitter right here. When it attached to a chemically gated sodium channel, my membrane charge went up. This increase in membrane charge, I would call this an EPSP, an excitatory postsynaptic potential. Neuron number 2 right here, its membrane charge got more positive. The membrane charge gets more positive. That's an excitatory postsynaptic potential. The same thing applies over here with an IPSP, an inhibitory postsynaptic potential. We're still talking about membrane charge because of potential. We're still talking about that neuron that receives the message with postsynaptic, but now we're talking about a change that is an inhibitory change. It's a change that makes us more negative. Here's that word we used on the last slide, hyperpolarization. So an inhibitory postsynaptic potential, an IPSP, is when I change my membrane charge and I make it more negative. An IPSP is a change in membrane charge that makes it more negative. When we're thinking about the way that a neuron works, let's look at a picture we ended with last time. Remind me what page this picture was on? Is it like page 9, Jacqueline's saying? Okay. Page 9 shows us the way that things normally work with a neuron. So a neuron's chilling out here at resting membrane potential. A neuron receives a neurotransmitter. When it received this neurotransmitter, it made its membrane charge go up. So the neurotransmitter that it received right here opened up a chemically gated sodium channel. It generated an excitatory postsynaptic potential where my membrane charge went up. I got excited. I'm getting closer to threshold. Maybe I'll get to talk to my neighbor. Now I get another chemical, another neurotransmitter that comes and attaches to a chemically gated sodium channel. More sodium comes in, I get more positive. It's another excitatory postsynaptic potential. My membrane charge went up. But then I received another neurotransmitter. This neurotransmitter made my membrane charge go down. So this neurotransmitter either opened up a potassium channel or it opened up a chloride channel. It did something to let ions in that made my membrane charge more negative. Because my membrane charge got more negative, we call this an inhibitory postsynaptic potential, an IPSP. So my membrane charge went down. I'm waiting a little bit, waiting. Hey, I got another message. This message made my membrane charge go up. My membrane charge went up. So this is another excitatory change. But I got another message, made it come back down. My membrane charge got more negative, inhibitory postsynaptic potential. This neuron, though, then received a neurotransmitter message that was so loud and so strong that it hit threshold. Remind me, when a neuron hits threshold, which kind of ion channels open up at threshold? Which kind of ion channels open at threshold? Yeah, threshold is the point where voltage-gated channels take over. Voltage-gated channels, voltage-gated, oops, spell it right here, voltage-gated sodium channels. This is the point where they take over. All of this stuff that we saw down here, all of this mess, all of this was because of chemically gated channels. I got a chemical right here, I got a chemical right here, a chemical right here. All of this stuff that we saw happening here, all of that was because of chemically gated channels. And neurons have chemically gated channels for multiple things. For sodium, for potassium, and for chloride. Make sure, as we're thinking about what makes my membrane charge go up, what generates these EPSPs, and what generates the IPSPs, makes my membrane charge go down. Make sure keeping track of which ions do each of those things. What are our thoughts / feelings about this? What questions do we still have? Okay. Yeah, so we're getting there, Christina. I promise. We're taking a roundabout way to that. Okay. Well, let's talk a little bit about the topic that Christina mentioned in her question. And the topic that Christina mentioned is something called summation. Summation. Summation is something that all of your neurons do. Neurons are never just receiving one message at a time. I actually feel like life right now in the pandemic is like such a good example of summation. I don't know about you guys, but I mean, you've heard my kids in the background, right? So I've got class going on, and I've got my kids going on, and I've got a whole bunch of things all going on at once, right? We've got the election going on. I've got lots of messages in my life that I'm mixing together to figure out what I should be doing at any point in time. Neurons are getting all kinds of neurotransmitters, all kinds of different chemicals that are being released toward them at the same time. Neurons are going to add together those messages to figure out how they should respond. Now, neurons really just have two different responses. Either they are going to talk to their neighbors, or they're not going to talk to their neighbors. That's really all neurons can do. They're either all in or they're all out. So when we talk about a neuron doing summation, the goal of summation is for a neuron to figure out, should I or shouldn't I talk to my neighbors? For a neuron to be able to talk to its neighbors, it's going to have to get to threshold. So the goal of summation, the goal of adding together all of the messages a neuron receives, is for it to get to threshold. If it's supposed to talk to its neighbors, it's going to get to threshold. If it's not supposed to talk to its neighbors, it's not going to get to threshold. So summation is a process a neuron uses to listen to the messages from more than one neighbor. So let me see which picture I want us to go to first. Here. We'll come back to that question. Here we go. Let's first talk about an analogy for what summation is like, because there's two ways that a neuron can listen to more than one message. A neuron can listen to more than one message all coming from the same neuron that it's talking to, or it can listen to more than one message coming from different neurons that it's listening to. So I can't take credit for this analogy. This actually came from a student. There are two ways. So say we're all being great, we'll pretend, right? We're going to pretend that we put our phones away during class. So after class is done, we pick up our phone and we see that we have 99 texts. 99 texts. I don't know about you guys, but my blood pressure would be like through the roof. Like, are you kidding me? What are all these messages? Two ways that we could have gotten 99 texts. Either all 99 of those messages came from one person and they just kept sending the same message over and over again, or we got added to one of those infamous group chats, right? So maybe we don't know everybody that's part of Flying Sombrero, but everybody in Flying Sombrero is having a great conversation over here. We got added to the group chat. We are getting all of those messages. Now, if they're talking about the best places to go pick up food, to eat at home, somebody might say, hey, I love McDonald's. Somebody else says, hey, I love Chick-fil-A. Somebody else says, hey, I love Chipotle. We've got lots of different ideas about where to pick up food. You could mix all of those ideas together from a whole bunch of different neurons, if you will, or a whole bunch of different people to figure out where you should go pick up food. Or maybe you got that one friend, in this case, Captain Marvel over here. I'll give Captain Marvel a pass. I'll let her text me 99 times, but just this once. This is not going to happen again. If she's over here and she keeps saying the same place, which if we're going to pretend Captain Marvel is my daughter, she calls it Old McDonald. So she would just keep saying, Old McDonald, Old McDonald, Old McDonald. That's what my daughter wants to eat. Okay, so I'm adding together all of the messages from one person telling me where to go versus multiple people telling me where to go. This is the two ways that neurons do summation. When we're talking about one person sending a message over and over and over again, this is something called temporal summation. Temporal summation. Now, in this case, when I'm using the word temporal, I am talking about time, temporal relating to time. So if one neuron sends me a bunch of messages over some period of time. I'm listening to one neuron talk a whole bunch, 99 messages from Captain Marvel. That's temporal summation, one neuron talking to me. More likely though, when we're thinking about with a neuron listening to more than one messages, usually they're getting those messages from more than one source. That's something called spatial summation. Spatial summation. Spatial summation, that first word spatial means space or distance, area maybe. I'm trying to think of a good way to describe it. Spatial summation is when more than one part of my neuron gets a message from different neurons that are talking to it, and it adds all of those messages together. So spatial summation, more than one neuron talks to me. Temporal summation, one neuron talks to me repeatedly. Regardless of whether it's one neuron talking to me or multiple neurons talking to me, I'm always going to add those messages together to figure out what the right response would be. So let's take this texting analogy and apply it to the picture from your packet. What page do we have these pictures on? Okay, page 7 and 8 kind of bleeds over across them. Okay. If a neuron were to just hear one message, which here's a spoiler alert, that's never going to happen. If a neuron were to hear just one message, that message comes and a neuron changes its membrane charge a little bit, but it doesn't hit threshold. So over time, those leakage channels and the sodium potassium pump get it back to normal. And then later it hears another message, and that makes its membrane charge go up a little bit. Again, still not enough for threshold. It's going to go back down. If a neuron's not hearing multiple messages at a time, if it's not doing summation, we might stay under threshold forever. We may never decide we have to talk to our neighbor. If we're doing temporal summation, notice how this one neuron who was talking to me sent me multiple messages. So while my membrane charge was already a little bit higher because I just brought in some sodium, I got another message that opened up even more of those sodium channels. So even more sodium came in, enough to get me to threshold. That's going to make me talk to my neighbor. So if I receive two messages from the same neuron, multiple text messages from the same person, that's temporal summation. Because the first one was an EPSP, it made my membrane charge go up a little bit. That makes it easier for my second one to make my membrane charge go up enough to get to threshold. And remember, with spatial summation, more than one neuron is talking to my neuron here. So I see a message coming in over on this dendrite. I see a message coming in over on this dendrite. Both of these messages come, and enough ion channels open that I hit threshold and I spike up and talk to my neighbor. I saw the comment in the chat. Yeah, thinking about spatial summation, think about how when you're in that group chat, people from all over the United States, different spaces, different places are talking to you. That's absolutely right with spatial summation. Different neurons talk to you at the same time versus temporal summation, one neuron talks to you repeatedly. So multiple neurons for spatial summation, one neuron for temporal summation. What questions do we have about spatial summation versus temporal summation? So Jacqueline asked, those messages come from EPSP and IPSP. Not quite, Jacqueline. The messages that one neuron is sending to another is in the form of a neurotransmitter. So in this case here, where I've got one neuron that's sending multiple messages, what's happening is this neuron is spitting out neurotransmitters multiple times. Those neurotransmitters came down, and at this point, they opened up some chemically gated channels. And then at this point, they opened up more chemically gated channels. So when we talk about IPSPs or EPSPs, those are the membrane charges. So here, let's make a list here. IPSP, EPSP. These things are the changes in membrane charge, the changes in membrane charge. The way I make those changes is by using neurotransmitters. So we have what are called inhibitory, oops, I'm going to spell this wrong, inhibitory neurotransmitters, and we have excitatory neurotransmitters. These are the things that are released by a presynaptic neuron. These are the actual messages. When those messages are received, they lead us to have IPSPs or EPSPs. They lead us to change our membrane charge in particular ways. So to say it again, neurotransmitters are the messages. When the messages are received, they change the membrane charge, either making an IPSP or an EPSP. Does that help? Okay. And Christina's question is 100% correct. Yes. The process of summation only happens until I get to threshold. So the goal of summation is to get my membrane charge to threshold, to get me high enough to be able to talk to a neighbor. Once my membrane charge is high enough for me to talk to a neighbor, I don't need to listen to messages anymore. And in fact, we're going to talk about on Friday, there's actually a period of time where this neuron like physically can't listen to messages. It's called the refractory period because it's too busy responding to the other messages. So summation is what a neuron uses to get to threshold. When I add together those messages, my goal is to get to threshold. I'm trying to get to that point where I don't have to listen anymore. I just get to talk to my neighbor. Once I hit threshold, summation is done until I'm back to resting membrane potential. Here, let me show you this picture really fast. We'll come back. When you think about a neuron that is trying to figure out if it gets to talk to its neighbor or not, consider that a neuron pretty much always is going to be doing spatial summation. It's pretty much always listening to more than one neuron. Some of those neurons are sending messages that are excitatory. So some of them are spitting out neurotransmitters that make the membrane charge go up. So neurotransmitters that help us make EPSPs, membrane charge goes up. But some of those neurons are sending messages that make the membrane charge go down. Some of these are sending inhibitory neurotransmitters. So when a neuron's trying to figure out whether or not it should talk to its neighbor, at the same time that some of these neurons are trying to bring it close to threshold, some of these other ones are trying to get it away from threshold. So when we think about how a normal neuron is functioning, rather than just seeing some positive messages that tell it to send a message to its neighbor, what's more likely to be happening is that a neuron gets some positive messages and then maybe it gets a negative message. And maybe it gets a positive and a negative at the same time. When we talk about summation, it's literally like math. Like literally we're adding a positive to a negative, to a positive and a negative. We're doing math where we start at negative 70 millivolts. Here's where we start. And then I got a message that is a positive 5 millivolts, say. I got an excitatory neurotransmitter. So when I got that message, that brought me to negative 65 millivolts. We're just doing math here. So then I get a message, and this is an inhibitory message. This message was negative 10 millivolts. I just generated an IPSP, an inhibitory message. Now I'm down to negative 75 millivolts. So we're literally, in summation, we're literally doing math. Here's where I started. I got an excitatory message. Okay, now I'm at negative 65. Awesome. I'm close to where I want to go. Oh, now I got an inhibitory message. That makes me more negative. I'm adding and subtracting and adding and subtracting. If in the process of adding and subtracting, I finally get to threshold, then I get to talk. But if I don't get to threshold, I don't get to talk. That's the way that summation works. I can't see your faces, so I don't know what we're thinking. Keenan's mind's blown. Yep. Christina's soaking it in. Yeah, Ariel's a little nervous. Yeah, so I like that way of thinking about it, Jacqueline. Yep, so a neurotransmitter message is sent, and that has an effect on the membrane charge. That effect is added together for all of the neurotransmitters in the process of summation, and summation is what we use to get to threshold. Yes, I like that. Yeah, so Nicole's asked clarifying question. Threshold is not the point when neurons talk to each other. Threshold is the point when a neuron realizes it's going to be allowed to talk to its neighbor. So if I hit the charge of negative 55, my voltage-gated sodium channels will open up. I can't talk to my neighbor until I'm at positive 30. But for me to be able to get to positive 30, I have to use voltage-gated sodium channels. And voltage-gated sodium channels won't open until I get to negative 55. So here, now that we've talked a little bit about summation, let's go back to this picture here. This is the point where voltage-gated sodium channels open. The only way a neuron can get all the way to positive 30 is if voltage-gated sodium channels open. So until I get to negative 55, I have no chance of ever getting to positive 30. Positive 30 is my charge when I'm talking to my neighbor. If I hit negative 55, it's a given that I'm going to get to positive 30. I got to get there first before I can talk. To get to negative 55 is where I use all of those chemically gated channels. Some of them make my membrane charge go up. Some of them make my membrane charge go down. My chemically gated channels, all of this right here, the summation process, that's those chemically gated channels. If I get to threshold, then I'm going to use voltage gated channels to get all the way to positive 30. Which is the charge that I'm actually able to talk to my neighbor at. Does that help? Yeah, go for it, Nicole.
So to use your analogy of your daughter, harassing you to take her to McDonald's, the ESP would be her saying McDonald's, McDonald's, McDonald's until you hit negative 55. Then at negative 55, you get in your car, you start your car. That's the voltage gated, and 30, you're at McDonald's ordering?
Sure.
Would that
Yeah,
be-
I like that.
that work? Okay.
Yeah, so all of the hints, come on, mom, let's go, let's go. Okay, fine, we'll get in the car. My voltage gated sodium channels take me over to get me to McDonald's. Yeah, I like that. Yep, so here's where I get my food. Here's where my daughter gets her Happy Meal. Put it that way. So here she's sending me the message, I want a Happy Meal, I want a Happy Meal. My husband's like, we had McDonald's yesterday. I want a Happy Meal, we had it yesterday. I want a Happy Meal. Okay, we're going. I like that. Yep, that's perfect. So Christina, when you ask about this graph, are you talking particularly about the one that you see right here, or are you talking about the general neuron potential graph? Okay, this one right here, I can't remember if it's on the exam or not. Let's pretend like it is, and we can talk about what we'd want to know on it in case it was on the exam. First thing we would want to know is those numbers that apply on any of our neuron graphs, right? So -70, that's our resting membrane potential. -55, that's our threshold. And +30, that's the value that I get to when I'm able to talk to my neighbors. That's the first thing we want to know on any neuron membrane potential graph. The second thing that's specific to this graph that we would want to know is we would want to know which kinds of ion channels could be used to make an EPSP or an IPSP. Now remember, these abbreviations here, I suggest you just learn them as an abbreviation, unless you want to know the words. That's totally fine. If you want to learn the words, I'll type them out for us here. EPSP. Excitatory postsynaptic potential. Here's our first one. That's what an EPSP is. IPSP, inhibitory postsynaptic potential. When we talk about EPSPs, excitatory postsynaptic potentials, these are changes in my membrane charge that are positive. These are making things go up. So one thing that we'd want to know on this graph is that all of these little green bumps where we get more positive, those are excitatory postsynaptic potentials. We'd want to know that. The red points where we get more negative, those are my IPSPs, my inhibitory postsynaptic potentials. So we want to know what happens when we talk about having an EPSP, membrane charge goes up, or an IPSP, membrane charge goes down. The last thing we would want to make sure we knew was which kinds of ion channels generated each of these things, or which kind of ion channels we probably opened at each of these points. When I generate an EPSP, an excitatory postsynaptic potential, which kinds of ion channels help me to make EPSPs? Who raises that membrane charge? Yeah, chemically gated sodium channels. Chemically gated sodium channels. So last thing we would want to know is that this change that I see right here could only be caused by a chemically gated sodium channel. This change right here could only be caused by a chemically gated sodium channel. Hey, this is an IPSP. This is an inhibitory postsynaptic potential. Which kind of channels could make this happen? Who could be responsible for pink? Yep, so we're still chemically gated and we got two options, right? Chemically gated potassium or chemically gated chloride. Yes, absolutely. So the final thing you'd want to know, if I put a little arrow that pointed right here, for example, or I put an arrow that pointed right here, I could ask you which kind of ion channels opened up at these points. You would see My membrane charge went down. So if my membrane charge goes down, that's either a chemically gated potassium channel or a chemically gated chloride channel. Or right here, my membrane charge went up. That's going to be a chemically gated sodium channel. That's my only option. So those are the things that you would want to know on this graph. So Nicole asked the question, since the charges that we're seeing on this graph are different, would we know that there's spatial summation happening? Yes, that's correct. And you can kind of see that on our picture up here too. Each neuron that synapses with another neuron can only ever send one message. It can only ever send an excitatory message or an inhibitory message. So the fact that I'm getting some excitatory messages and some inhibitory messages, we know that we're doing spatial summation. I'm getting more than one kind of message. So absolutely, this is definitely an example of spatial summation. Let me bump really fast to this picture here. Can you help me out? What page are we on in your notes? I jumped ahead here. Okay, this is page 11. So this is one of the last things that I want to land on today, since we're getting low on time. Here's what why I want to highlight this picture. This picture shows me my presynaptic neuron. That's the neuron that's doing the talking. And it shows me my postsynaptic neuron, my neuron that's doing the listening. When we talk about neurons sharing messages with each other, those messages are these little neurotransmitters I see right here spitting out. So I spit out neurotransmitter messages, and they cause us to open these chemically gated ion channels. We just talked about how a neuron does summation, how it listens to a lot of different messages at once. Perhaps when I see this channel right here, this chemically gated ion channel, maybe this is a chemically gated sodium channel, but recognize that right next door to it might be a chemically gated potassium channel, or we might have a chemically gated sodium channel next to it. When we think about, this is just one dendrite on a neuron over here. When we think about the way that neurons talk to each other, or that they listen to each other, we've got this synaptic cleft space where more than one presynaptic neuron might be talking to the postsynaptic neuron, might be releasing neurotransmitters. So lots of different chemically gated ion channels all across the membrane, all across the dendrites of a neuron. Their job is to receive all the different kinds of messages that a neuron is getting and mix them together, the goal being to ultimately open up voltage-gated channels. So here, let's see. Let's do this. Let's end with this, because we've talked about these things some today. What page is this neuron on? We'll end with this neuron here. Okay, we're on page 12. So we've already said a bunch of this stuff, but we're going to go through and we're going to clarify to make sure we know. First question, my favorite question when it comes to channels. When I'm talking chemically gated channels on a neuron, what opens the chemically gated channels on a neuron? How do I get those open? Chemically gated channels. Yeah, exactly. Okay. Chemically gated channels only open with the right chemical. And when we're talking about neurons, the chemically gated channels open with neurotransmitters. When you're trying to think about on a neuron the places where I would have chemically gated types of channels, the only place on a neuron's membrane that's going to have chemically gated channels are the places on its membrane where it's going to receive chemicals, or the places on its membrane where it's going to receive neurotransmitters. Where did we talk about at the very beginning of class being the place that neurons receive messages? Where do neurons receive messages? Yeah, so the place on a neuron that I'm receiving my messages are the dendrites. Here's like an underline highlight star idea. Chemically gated channels, whether it's chemically gated chloride channels, chemically gated sodium channels, or chemically gated potassium channels, all of these chemically gated channels, the only place on a neuron that you're ever going to find chemically gated channels are the dendrites, because that's the only place on a neuron that's ever going to receive neurotransmitters. So my places where I've got these channels, I'm just going to kind of draw a line around parts of my dendrite here. This looks real terrible. All of these places here that I'm tracing in blue, all along those dendrites, that's going to be the places on my plasma membrane where I have these chemically gated channels. Chemically gated channels. I need chemicals to open them up. The only place we got chemicals is at the dendrites. That's where I'm receiving them. Now, I didn't have you label this before, so let me ask if we recognize it here in the chat. What's this part of a neuron called right here? What's that part of a neuron called? Does anyone remember? When I drew the picture for you, I drew it as a triangle. Yeah, so this part right here is called the Axon Hillock. The Axon Hillock. When you think about the Axon Hillock, I want you to think about that the function of the Axon Hillock is to do summation, to use a word that we just now talked about in class. The Axon Hillock is the location on a neuron where we do summation. The Axon Hillock is the place on a neuron where it does summation. When I do the process of summation, my goal is to get to threshold. Because what happens at threshold? What did we say happens at threshold? I promise it's not a trick question. What happens when a neuron hits threshold? Yeah, my membrane starts to depolarize. Voltage-gated channels open, okay? Voltage-gated sodium channels, yeah, in particular. When I hit threshold, voltage-gated sodium channels open up, which means the Axon Hillock is going to be the first place where I see voltage-gated sodium channels. I'm doing summation. I'm adding together all of the messages that I got here, all of my little positives and my little negatives. I'm adding them together right here at the Axon Hillock. If I get to -55 at the Axon Hillock, when I add everything together, I'm going to be able to depolarize my membrane. I'm going to be able to make my membrane positive. Now, we'll talk about this more on Friday, but if you remember from our muscles chapter, we talked about a thing called an action potential. That was where I change my membrane charge in one place, and then I send that change everywhere in my cell. When we talk about the way that a neuron works, they do action potentials too. So I first Let me label it for you. Voltage-gated sodium channels. I first open voltage-gated sodium channels at the Axon Hillock because I hit threshold right here. Once I hit threshold right here, that's going to make the neighboring places on my membrane get more positive too. And they're going to hit threshold. And that's going to make the neighboring places on my membrane get more positive. And they're going to hit threshold. And we go down, we send our action potential or our change in membrane charge. First, I opened up the voltage-gated sodium channels here. That leads me to open up the voltage-gated channels all the way down my axon, all the way down to my axon terminals. I'm using voltage-gated sodium channels to depolarize the membrane. We didn't talk a lot about it today, but when that positive charge, when the positive 30 gets all the way down to my axon terminal, down here is the one place on my neuron where I find voltage-gated calcium channels. So I'm going to circle these spots here. The one place on a neuron where I have voltage-gated calcium channels. Oh, that was terrible. Here comes a really terrible circle, guys. Voltage-gated calcium channels. Voltage-gated calcium channels are down here. When my membrane charge gets to positive 30 down here at the end, these channels open up and bring calcium inside the cell. Remember, calcium comes in and the neurotransmitters peace out. They can't even with calcium. So this is where I bring calcium into my axon terminal to spit out my neurotransmitters. And Jacqueline is 100% right. The last kind of channel that we don't have labeled here yet are those voltage-gated potassium channels. The job of voltage-gated potassium channels is to repolarize the membrane after the voltage-gated sodium channels depolarize it. So the last thing we're going to add to our picture here of the neuron are those voltage-gated potassium channels in all the same places as our voltage-gated sodium channels are, because their job is to repolarize the membrane after we've changed it completely, after we've completely freaked it out. So let's summarize here in closing. When a neuron receives a message, that message comes in the form of a chemical. Those chemicals open these chemically gated channels that I find all over the dendrites of this neuron. The sum of all of those chemical messages, the pluses and the minuses, is added together. We put it in a big blender here called the Axon Hillock. If we get to threshold at the Axon Hillock, voltage-gated sodium channels open up from here all the way down the axon, all the way to the axon terminals. When we depolarize the membrane at the axon terminals, that causes us to open voltage-gated calcium channels. When it's positive 30 down here, those ones open up as well to allow neurotransmitters to spit out. But we don't want to stay at positive 30 anywhere on this membrane. So to help us get back to normal, voltage-gated potassium channels will repolarize the membrane all the way down, everywhere that we depolarized it with sodium. That's all I want to cover today. I don't want to dive into anything new. Before we call it a day today, do we have any specific questions we can put to words right now? I know we're all soaking it in. It's a lot. As we wrap up, quick reminder that we're not going to have a session tomorrow morning. It's going to be Friday morning. So tomorrow morning, nothing going on. Work on trying to wrap up the packet. Try to do at least one attempt on this lecture homework to see what questions you still have. We will wrap up lesson number 10 on Friday. So we'll cover the action potential. We will cover the ways that a signal goes down the axon here. We'll review anything that you still have questions on. So plan to bring those questions. As always, feel free to reach out via email. Otherwise, I hope to see most of you all on Friday morning. So Friday morning at 9:30.